Hello, everyone. Thank you for attending our virtual presentation of the Evolving STEM program. My name is Abigail Matella, and I am a postdoctoral researcher at the University of Pittsburgh. I work in the laboratory of Dr. Vaughn Cooper, who developed this incredible outreach program to educate students with engaging learning experiences that motivate their interest in science. For the past 2 years, I have worked to refine and expand Evolving STEM to serve diverse student populations. I am joined by 2 talented educators that have implemented the Evolving STEM curriculum in their classrooms and labs.
Hi everyone, my name is Edwina Kinchington and I teach life science and biomedical technology at the Pittsburgh Science and Technology Academy in the city of Pittsburgh. I have used the Evolving STEM curriculum for the last 2 years with both my 10th and 11th grade students.
Hi everyone, my name is Ron Kinzer. I teach life science at Wrinkly Academy, which is just outside of Pittsburgh. I've used the Evolving STEM curriculum for 3 years now. We are very excited to share this experience with you as it's an exciting and effective way to teach high school students about evolution.
The Evolving STEM curriculum was created and developed by Dr. Vaughn Cooper and his lab at the University of Pittsburgh School of Medicine in collaboration with high schools in New Hampshire and the Pittsburgh area, as well as the Center for Evolutionary Biology and Medicine. After this presentation, please feel free to take a closer look at the curriculum resources in our Google Drive folder and visit our website, EvolvingSTEM.org, to learn more about our program. The primary driving force behind the Evolving STEM curriculum is to help students develop an accurate and complete understanding of how evolution works, with the hope of creating a scientifically literate society and a well-prepared STEM workforce. Evolving STEM achieves this by providing an engaging and authentic hands-on scientific experience. Typical evolution curricula around the country rely heavily on passive learning strategies, including lectures and textbook readings. One of the most recognizable examples of how evolution can lead to adaptive change is Darwin's finches and beak adaptation in response to food sources. Of course, the problem with this is students would never be able to see a finch population change in a classroom setting. On the other hand, when bacteria are grown under strong selection for attachment to surfaces, populations evolve to be better at attaching to these surfaces by forming copious biofilms, and high biofilm forming mutants often have visibly distinct colony morphologies like those shown here. The Evolving STEM curriculum recognizes the effectiveness of student-centered, active learning and focuses on leveraging student engagement to promote a deeper understanding of evolution. So what exactly is a biofilm? A biofilm is a surface-dwelling community of microbes encased in a protective coating of self-produced polymers. Biofilms are the dominant form of microbial life. They are also structured, heterogeneous environments that include varied ecological niches. Bacterial populations diversify to colonize these niches, and advantageous mutations often result in a variety of visible differences in colony morphology from the ancestral genotype. 4 steps of biofilm formation should be highlighted here, as they are integral in the Evolving STEM experiments. In step 1, bacteria attach to a surface. In step 2, a biofilm is formed as more bacteria accumulate and produce an extracellular matrix. In step 3, bacteria disperse from the biofilm, and in step 4, bacteria can recolonize new areas to repeat the life cycle. Adaptation to the biofilm lifestyle occurs within days, requires little specialized equipment, and can be offered in any classroom laboratory with a sink. Students work with the plant growth promoting bacterium Pseudomonas fluorescens, which has several qualities that made it a good candidate for use in a high school classroom. First of all, it's benign and thus safe for students with no microbiology experience. Secondly, is the subject of a large body of research on its capacity for rapid and conspicuous adaptive evolution and biofilm-related conditions, so we know it will make an excellent model organism for the questions we are asking. Lastly, experimental evolution studies find a high frequency of adaptive mutants with novel colony morphologies in less than 2 weeks, which is ideal for high school labs working with time limitations. Here we can see scanning electron microscope images of a single bacterium that is attached to a surface. We can also see an entire biofilm community. Pseudomonas fluorescens get their name from their ability to fluoresce under UV light, shown here. First, we'd like to provide an overview of some of the key aspects of the evolution in action experiment. The beauty behind the Evolving STEM experiments is that we can replicate the biofilm lifestyle using basic equipment that most high school labs have. Basically, we can create the opportunity for biofilm formation and bacterial dispersal and recolonization using a simple bead transfer. The experiment tests how populations of Pseudomonas fluorescens change over time when we select for bacteria that are best suited to life on the bead. This method selects for bacteria that can complete the entire life cycle. Bacteria must stick, swim, and recolonize beads. We compare bead populations to control populations and to ancestral populations. The control population is a sample of free-swimming or planktonic bacteria. To identify changes, we must make observations of biofilm growth in the test tubes and the colony morphologies of bacteria growing on agar plates. So there's a nice combination of simple and modern hands-on biotechnology skills being put to use by students in a straightforward assay. One of the greatest aspects of the Evolving STEM curriculum is that it encourages students to think about experimentation in a quantitative way. On the first day of the experiment, a single cell will multiply to 10 billion cells. Students know mutations are rare. Around 1 in 1,000 cell divisions produce a mutation. Students sometimes fail to consider, however, that given the number of cells and how quickly bacteria like Pseudomonas fluorescens can divide, we will still see an abundance of mutations at random in a short time. The population will start out as genetically identical. This ancestral genotype is indicated by the black-colored bacteria shown here. As mutations arise in the population, however, here indicated by the different colored bacteria, the population will quickly diversify. Doing some simple math, students can see that millions of mutations arise in their bacterial populations each day. That's 10 to the 10 bacteria times 10 to the minus 3 mutations, equaling 10 to the 7 mutations per day. In addition to encouraging quantitative thinking, these experiments allow students to practice abstract reasoning. Students will understand that although mutations are random, the selective pressure is not. Of the previously mentioned 10 million mutants, some of them may have acquired a mutation that allows them to better survive and reproduce on the bead than their ancestors. These mutants, represented by the yellow bacteria, shown successively here, are better adapted to the biofilm life cycle. Bacteria with beneficial mutations will therefore increase in frequency in the population over time, as shown in this picture. The Evolving STEM experiment allows students to see this. There are two places where biofilms are formed during the experiment. As previously stated, the bead is the selective force that collects bacteria that are increasingly more adapted to forming biofilms. This, however, is not easily observed by the eye. Bacteria also attach to the walls of the test tube at the liquid surface, that is, the air-liquid interface shown here and here. As high biofilm forming bacteria increase in frequency in the population, students will be able to see increased biofilm formed at the air-liquid interface within each test tube, again as shown here. There are two platings performed during these experiments. Control and experimental groups are plated early on to show students what the populations look like after a very short period of selection. When bacteria are plated, cultures are first diluted, then spread across the agar plates. Individual bacteria will attach to the agar and replicate exact copies of themselves to form visible colonies, again shown here. During this early plating, colonies from both control and experimental populations should look smooth, like the ancestral population. However, after 4 days of selection on the bead, the experimental population will have an increased frequency of biofilm-adapted mutants that are visibly distinct from their ancestors. These mutants, commonly display colonies that are smaller or larger than ancestral colonies with a wrinkled appearance like those shown here. We know that these changes to colony morphology are heritable because when we take samples of the individual colonies that show morphological change and grow them in a liquid culture, we see that all offspring of a wrinkly parent display a wrinkly colony morphology when plated. This helps students grasp the importance of genetic mutation leading to phenotypic change and adaptations in populations through selection. I will now turn it over to my colleague, Dr. Edwina Kitchington, who will discuss the application of the Evolving STEM curriculum in her class and lab.
Thank you, Dr. Kinzer. I teach at a science academy in the urban district of the Pittsburgh Public Schools, and I'm going into my 4th year with Evolving STEM. Here you can see Jay Brown left and Isabel Walker, both 16, working together on the Evolving STEM experiment in my lab classroom at the Pittsburgh Science and Technology Academy. More than 2,000 students have worked on the Evolving STEM curriculum, which is being implemented in 11 schools across 4 states. The Evolving STEM curriculum is leading to measurable increases in students' knowledge, as well as excitement about learning science and participating in STEM careers. Of particular note was the significant improvements in these areas for female students and students of underrepresented minority groups. Feedback from students has indicated strong engagement in the process of science and the understanding of evolution unfolding before their eyes. The Evolving STEM curriculum and experiments can now be acquired as a kit. These hands-on experiments can be performed for less than $10 a student. The Evolving STEM Evolution in Action experiment provides endless opportunities for question-driven extension experiments. As an example, you can see some of that in this slide. Students can come up with their own questions on the biofilm production ability of different evolved and/or ancestral colonies and test their predictions with a commonly used assay that quantifies biofilm production. We do this by growing cells overnight, staining them with the dye that attaches to the biofilm, and you can see that here in this plate, and by quantifying the amount of biofilm produced with a spectrophotometric machine that measures light absorbance, and that's shown here. We expect that biofilm adapted mutants will produce copious amounts of biofilm and thus stain very strongly. Students can analyze the data and quantitate the amount of biofilm production and correlate that with each mutant colony. Students have many opportunities to develop their own research questions, collect and analyze real-world data that they have created, and then make sense of that data through analysis. If you would like to learn more about the extension activities, please join our Zoom after this presentation. Here are some pictures of students performing the experiment in the classroom. The Evolving STEM program engages both lower level and higher level thinking students at various stages of learning. Every student that has worked with this module has stated they'd loved the hands-on experience, loved that they could drive their own questions, and they felt challenged to think and learn skills that pushed them. The only drawback was the smell from the Pseudomonas fluorescens. I will continue using this module and can now establish extension projects where students have continued to pursue their questions from the initial experiments for science fair modules. Next, Dr. Abigail Mutella will talk about a newly developed bioinformatics module based on the Evolving STEM experimental results.
Thank you, Dr. K. Now I will detail an extension that builds on the initial laboratory experiment by walking students through the process of using modern bioinformatics techniques to identify the genetic basis of the evolved mutants. This activity answers the question of how do evolved wrinkly mutants differ genetically from their smooth ancestors, tying together the connection between phenotypic evolution and its basis in adaptive mutations. To identify the mutations that underlie wrinkly mutant phenotypes, the first step is to extract DNA from a wrinkly mutant, then sequence the mutant's genome and use bioinformatic methods to compare the mutant's DNA to that of its ancestor. This method allows us to identify adaptive mutations that had arisen during selection for high biofilm formation. The Cooper Labs Microbial Genome Sequencing Center used this method to analyze data from mutants that evolved during experiments performed by high school students, including students from 2 Pittsburgh schools, SciTech and Alderdice. Our lab extracted DNA from 10 mutant bacterial colonies and used a technique called Illumina sequencing to determine the sequence of DNA base pairs in the mutant's genome. In short, this technique works by cutting the DNA into tiny fragments, which are attached to a glass slide called a flow cell. The fragments are amplified to produce identical clusters of each fragment. Then each nucleotide base of the fragment is labeled with a fluorescent tag. Fluorescent tags are excited by a laser, and their emitted signal is captured by a camera. The process of labeling and signal capture are done sequentially along each fragment to identify its DNA sequence. The sequencing data is stored in a FASTQ file, a text file that contains a sequence identifier, the sequencing reads, and quality scores. This is big data, as these files can be hundreds of megabytes in size. Each sequence is approximately 150 base pairs long, but the entire Pseudomonas fluorescens genome is approximately 6.7 million base pairs long. This is where bioinformatic methods will help us to reconstruct the mutant genome by stringing together the small, overlapping DNA fragments and comparing them to the ancestral genome. This is kind of like putting together the small pieces of a puzzle to see the entire picture. To do this, we will use the bioinformatic tools available in Patrick, a free service that allows us to run the software online, eliminating the need to install or run these programs on your computer. Students will be able to run these analyses themselves. In the process, they will learn how to quickly analyze the massive amount of data produced during genome sequencing to identify adaptive mutations. I'll now walk you through a short video that shows how to perform bioinformatic analysis with Patrick. First, navigate to the Patrick website and log into the EvolvingSTEM account using the username and password provided in the Google Drive Documents folder. From the home page, click on the Workspaces tab, then click on Workspaces at the top of the drop-down box. Now double-click on the EvolvingSTEM folder. You will see 10 folders that contain pictures and sequencing reads for each mutant. Representative images of each mutant and an image of the Pseudomonas fluorescens ancestor for comparison are also provided in the Google Drive document folder. To run the analysis, click on the Services tab. Then select Variation Analysis from the drop-down menu. Load the reads belonging to your selected mutant by clicking the folder icon next to Read File 1. Then navigate to the Workspace folder where your files are located by double-clicking the parent folder, double-clicking EvolvingSTEM, double-clicking the folder of the mutant that you want to analyze. In this example, I'm analyzing mutant number 1. Now, you want to click on the read file with the extension .fwd paired.fastq.gz and then click OK. We'll then repeat these steps for read file 2, but instead load the read file with the extension .revpaired.fastq.gz. We'll now set the parameters for the analysis by selecting the Bowtie 2 aligner and FreeBayes snip collar. Bowtie 2 is software that takes the short reads generated during sequencing and matches them to a reference sequence. FreeBayes compares these mapped reads to the reference and identifies any differences between the two. These differences are the mutations present in your mutant. Select Pseudomonas fluorescens SPW25 as your reference sequence by typing its name into the target genome box. Then choose the first option from the drop-down menu. Click the folder icon next to the output folder box to choose the folder where you want the output to be sent. I will choose the practice folder for my output. Now you'll need to name your output. Click the arrow in the Paired Reads library to place your reads into the Selected Libraries box, and then click Submit. You can click on the Jobs bar to view your progress. The analysis should take approximately 5 to 20 minutes to run. Once the job is complete, you'll double-click on the job to open the output folder. This folder displays information about the job and the files generated by the analysis. The file we are interested in viewing is the all.var.tsv, which is a text file that will reveal the mutations that were identified in the mutant you analyzed. Double-click on the file name, then click the download icon to move the file to your home computer. You can open the .tsv file in a spreadsheet program, such as Excel. That's what I will be using here, to organize your data. The Drive folder also provides instructions for using Google Sheets. The TSV file separates column data by tabs. Excel and Google Sheets can use this information to place your data into appropriate columns. Although the free Bayes variant caller does a good job at identifying mutations, we will need to do some manual filtering of the data to identify mutations that occurred during the evolution experiment and we have high confidence are accurate. First, we will remove mutations that were already present in the Pseudomonas fluorescens ancestor used in the experiments. They differ from the published reference sequence we used in the analysis, but are not new mutations. To do this, I will delete 3 variants. The first at position 3,447,980, the second at position 985,332, and the third at position 45,877. Next, we will remove low-frequency variants. Because we are analyzing clones, we expect the genetic information to be identical across all of the sequenced reads. Variants found at less than 80% are likely errors introduced during sequencing or read alignment, so we should remove them. To make these variants easy to identify, we will first sort the data by highlighting all data columns. Then click Data, Sort, and sort by variant fraction from largest to smallest. I'll now delete all variants at lower than 80% frequency, which can be seen in column H. After removing low-frequency variants, you may need to remove variants with low quality scores, as these are unreliable. Again, highlight the data columns, then sort by score from largest to smallest, and delete all rows with a score of less than 200. Score values can be found in column F. Your data is now filtered, and the remaining rows identify the mutation or mutations that were present in the involved bacteria that you analyzed. I'll now walk you through how to interpret this output. Because there are 2 rows of data, we can see that the example was a double mutant. This means that it had 2 mutations in comparison to the ancestor. Column C tells us the nucleotide position where we would locate the mutation in the Pseudomonas fluorescens genome. For example, the first mutation was found at position 1,359,570. Columns D and E tell us how the nucleotide sequence changed. In this case, a single nucleotide was mutated from a G to a T. Columns J and K tell us how the mutation changed the codon sequence present at this position. In this case, a GAA is now a TAA. Column M tells us the effect of this change on the amino acid sequence of the encoded protein. In this case, the ancestor had glutamic acid at this position, but the evolved mutant has a stop codon. In some cases, Patrick will provide information about the gene's name, which is found in column Q, and proposed function, which is found in column R. In most cases, the gene will not be identified in the Patrick output. Column P provides a unique identifier for the gene that was mutated. This identifier can be used to find out more information about the mutated gene. A good resource for this is the Pseudomonas Genome database. Instructions are available in the Google Drive materials that explain how to use this database to find out the name of the mutated gene. Once you have finished analyzing all 10 mutants, you will find that mutations occurred in one of 4 genes, WISP-A, WISP-F, YFIR, or MOR-A. Some clones were double mutants and had either 1 mutation in 2 different genes or 2 mutations in the same gene. All of these mutated genes are members of 3 pathways that are involved in regulating a secondary signaling molecule, Cyclic-di-GMP. The mutations are all predicted to increase levels of this molecule so that mutants have more Cyclic-di-GMP present than their ancestors. These mutations are beneficial to biofilm formation because Cyclic-di-GMP is the central governor of the bacteria's decision to stick or swim. When levels of Cyclic-di-GMP are high, bacteria will attach to surfaces and produce biofilm. Although the students work with a safe bacterium, their results are medically relevant. A close relative of Pseudomonas fluorescens is Pseudomonas aeruginosa, a major opportunistic pathogen. Pseudomonas fluorescens and Pseudomonas aeruginosa share similar genetic pathways important for regulating Cyclic-di-GMP, including the WISP pathway. Moreover, high biofilm-forming Wrinkly mutants often evolve during Pseudomonas aeruginosa infections. Biofilm-associated infections are a major medical issue. They increase mortality, induce pro-inflammatory responses, and enable tolerance to antibiotics. The Cooper Lab recently published a study where high biofilm-forming Wrinkly mutants rapidly evolved in burn wounds infected with Pseudomonas aeruginosa. Whole genome sequencing revealed that the mutations that caused the high biofilm phenotype were exclusively found in WISP-A and WISP-F, 2 genes that also increased biofilm formation and caused Wrinkly phenotypes in the Pseudomonas fluorescens classroom evolution experiment. WISP mutants are also known to be very common in chronic airway infections of people with cystic fibrosis. Thank you for attending our talk. We hope you're interested in our Evolving STEM program and excited to use these materials in your classroom. Please feel free to take a closer look at our Google Drive folder to explore the detailed curriculum resources we have developed and visit our website to learn more about our program. We are happy to answer any additional questions you may have in the Zoom chat following this meeting. Thanks again.